this should be the lecture for the topic 4, right? Topic 4, yeah, part 1. So in part 1, we will talk about the cell division, and yeah, that will be the easy lecture on the mitosis and also the meiosis. Very easy lecture because it is something we have done before in the secondary school. Okay, the process would be the same. The detail would be the same. Actually, yeah, whatever you memorize in the secondary school is actually the same. Okay, so the only thing that's different is, of course, we will go slightly a bit of detail on the cell cycle, on the binary fission, which is something new to you maybe. Okay, and for the mitosis and meiosis process, everything will be the same. It's just that now it is more onto your understanding, your concept. Okay, not the description of the process. Because last time in secondary school, basically you just have to memorize. And then go into exam, you don't care, you just write everything. And then no matter what, you will still get the mark. Okay? But now in foundation, there won't be, not to say won't be, if let's say it come out, then you all will be very happy. Okay? But most of the time, the question won't ask you to describe the process of mitosis, describe what happened in the prophase 1. No longer this kind of question. This is too simplistic. Okay, so that's why no, right? Most of the time, the question that will be asked, it will be more onto if this statement is correct. Okay, what is being separated? So you have to be able to differentiate chromosome, homologous chromosome, sister chromatid. Which one is being separated at which phase? Okay, so that is the key. That's the key. So that's why you don't just memorize. You need to understand it. Okay, you need to know. Okay, what happened here and why it happened. Right? That is the key. Yeah, when you actually study this mitosis and meiosis. Okay, so that would be all that is all about this. Yeah. So, but don't worry. Yeah, don't worry. I am going to summarize everything. Okay, so you won't. I know how to. Okay, so now for this part 1, yeah, it will be purely on the mitosis meiosis. Okay, so this would be the learning outcome. Yeah, so let's see. So why I say it's very simple, because you look at the mind map. Yeah, it is very simple. Okay, so first part, yes, we will talk about the structure of the chromosome. And this one maybe is new to you. Yeah, maybe it's new to you. Okay, so the most basic unit of the chromosome would be the nucleosome. Yeah, nucleosome, that's the most basic unit. Okay, then done with the structure of the chromosome, then next we'll look at the cell division. So cell division, we learned that in prokaryotic cell, there will be using the binary fission only. Okay, and eukaryotic cell, they will use a more complicated cycle, we call it cell cycle. So cell cycle, there will be G1, S, G2, and the M phase. M phase can be mitosis or meiosis. It depends on what the cell wants to do. If the cell want to, increase in their number or the growth, okay, or for the repair and the replacement of the wear out cell, that one they will use the mitosis. But if let's say the cell is going to do sexual reproduction, it's going to do produce the gamete, then they will use meiosis. Okay, so that's the only difference. But this is actually just one phase under the cell cycle. Okay, so that's the key, and that is basically all the phase for mitosis and meiosis, and that's it. You just have to know what happened here, what happened here, and what happened here. Okay, you need to understand it. Then you need to understand the phase. Okay, that means that, yeah, that's it. Yeah, that's it. Okay, so it's just that now in the secondary school you memorize so many things for all the process here. What happened in prophase, what happened in prometaphase, what happened in metaphase, and then blah blah blah. All these things. Okay, so what is the important thing? When they ask for understanding, or when they use the graph to analyze it, okay, so how they can ask or what they are asking at. Okay, so most of the question that related to understanding, most of the question that related to understanding or test your understanding, or the higher order thinking questions for this part, well, normally they will ask for three things. Normally it's three things, okay? So they will ask for chromosome behavior, they will ask for the spindle fiber behavior, they will ask for the nucleus behavior. That is the three things. Okay? And in other words, this is the three things that you need to understand. You need to build the concept on. Okay? So don't worry, yes, I have actually summarized it in the table like this. So these will help you to organize your memory and also help your understanding and prevent the confusion. Because a lot of students will confuse, the process in the prophase 1 and prophase 2. Some students they will tend to confuse. Okay? So to prevent the confusion, yes, this is the one that can help you. Okay, so don't worry, later we will do it together. Yeah, we'll do it together one by one. Yeah. So as for now, I don't zoom in first. Yeah, I don't want to scare you first. Okay, so now this would be all the learning outcome. That's it. Very simple. Okay, then now we move on to the note. Yeah. So the first part we will talk about the structure of the chromosome first. Then we will go through the binary fission, then the cell cycle, then only then we look through the all the phase in the mitosis and also the meiosis. All right. So we look at it one by one. Yeah. So first thing first is the concept as I mentioned. The first one, prokaryotic cell and eukaryotic cell, we have learned that. Prokaryotic cell, it is going to divide by binary fission because they are too simple. Later you can see it's so simple that only 3 steps. Okay, well compared to eukaryotic cell, you will need to use a more complicated one, which is mitosis or meiosis. Reason, because they have nucleus. Right? They have nucleus, they have membrane-bound organelle to be separated, and also one more thing is that we will have more than one chromosome. We have more than one chromosome. Compared to the binary fission, the prokaryotic cell, they only have one nucleus. And some more, the nucleus is not a true chromosome also. So they can simply replicate and separate them. But for us, for eukaryotic cell, no. Eukaryotic cell at least must have 4 chromosomes. At least. Yeah, at least 4 chromosomes. It won't be lesser than that. Lesser than that normally is not a eukaryotic cell. Okay? So that's why more complicated. So that's why we have to use the more complicated process. Okay, so now you can see that here, eukaryotic cell, we will do the division of the cell nucleus, which is the mitosis or meiosis. Okay, then we have to do the cytokinesis, which is division of cytoplasm. So you have to have this concept is that mitosis and meiosis, only eukaryotic can do that. Why? Because mitosis and meiosis itself is actually the division of the nucleus. It is a division of nucleus. So prokaryotic cells don't have nucleus. How are they going to do the division of the nucleus? Okay, so after the mitosis and meiosis, then it has to follow by the cytokinesis. That only then one cell can become 2 cells. So this one is separate the nucleus, this one is separate the cytoplasm. That only then it can become 2 of the cells. Okay, so that would be the just a brief introduction. Okay, so now we look at the structure of the chromosome. Now we look at the structure of the chromosome. Okay, so well, first thing first, you have to understand DNA and gene are different things. DNA is actually a long polynucleotide chain. It's a long polynucleotide chain. So with ATCG, ATCG, AGA. So that is the sequence. That is the DNA. Okay, so DNA in the eukaryotic cell is always one long strand. That's always linear. Okay, so on the DNA itself, there are certain coding. So this part, let's say this is the sequence. Let's say this is the sequence that it code for, let's say, hair color. Then maybe over here, there's a sequence that code for the whether straight hair or curly hair. Then maybe over here, another sequence that code for maybe other protein or maybe insulin. So that means that the sequence on the DNA itself, the sequence that code for the protein, we call it gene. Okay, we call it gene. So there will be so many genes along the DNA. Okay, and of course you can see in between the gene, there will be the DNA that code for nothing. So this one is the so-called rubbish DNA. But in exam, don't write rubbish DNA, and this one is actually the intron. Okay, so don't write that. Okay, this one we will talk about this in the transcription. Yeah, in transcription only we will talk about that. Yeah, in the next lecture. Okay, so as for now, yes, this is a rubbish DNA basically. They don't code for anything. Yeah, they don't code for anything. Okay, so this long strand of the DNA contains a lot of the gene, right? Okay, So this DNA, it will be coiled and condensed so that it becomes chromatin. Then later on, the chromatin will further condense and coil during the mitosis or meiosis to become the chromosome. So DNA, gene, chromatin, chromosome are all different things. You have to remember that. Okay, it's a different status, yeah. Now we look at so that means that one strand of DNA is going to coil to become one chromosome like this. Okay, so how many chromosomes we have for human? Human beings? Any human beings should know, right? All the human beings should know how many chromosomes you have. 46. You have 23 pairs of chromosomes. So that means total 46 chromosomes. In other words, we are having 46 strands of this DNA. We are having 46 strands of the DNA. So that's why when during the mitosis or meiosis, they will condense and become 46 chromosomes. One chromosome means one strand of DNA. We have 46 strands of DNA. Okay, so you can imagine how many genes are there. So total 46 strands of DNA, right? So there are so many genes, right? So all of these genes that is needed in order to code for one organism, we call it genome. And we call it genome. Okay, so that's the meaning. Right, so yeah, this is a gene. Gene is just a sequence that code. So there will be sequence that don't code. Okay, then this whole thing is a DNA. So DNA will coil and condense to become the chromatin. So how they coil and condense? They will wrap it around the histone protein to become the chromatin. So after becoming chromatin, they will stay inside the nucleus in the nuclear plasm. Then whenever mitosis or meiosis occurs, then they will further condense to become the chromosome. So that means that when the cell is at rest, they are not dividing. It is actually occurs as chromatin. When they want to undergo prophase, then only then they will condense to become the chromosome. Okay, so that's the concept that you need to have, yeah? All right, so since we have so many chromosomes, we have 46 chromosomes. 46 chromosomes means that 46 strands of DNA. All right, so every single cell in us, except the gamete, every single cell in us, no matter your hair cell, your skin cell, or whatever cell is that, it will contain 46 chromosomes. That means 46 strands of the DNA. And you know how long is all of these strands of DNA? That means that, let's say you take out this 46 strands of DNA and then you combine them together for one single cell. You know how long is that? This long, 1.8 meter. All right, that's that long. So you can imagine you need to squeeze that long of the DNA into one nucleus. How small is the nucleus? It's so small. Yeah, every single nucleus will have 46 strands of DNA. And if you combine, it's 1.8 meter. So how are you going to squeeze that into the nucleus? So that's why the DNA, you cannot just simply plug that and dump inside. You cannot. It needs to be fully organized. It needs to be highly organized. That only then you can squeeze this 1.8 meter of the DNA into the nucleus. Alright? Every single cell will have it, yeah? Not just. So that's why you can, I think you have heard that before. With three, I just need three of your hair, I can clone you. It is true that it's not a myth. I just need to find get three hair from you, then I can clone you. Because one hair is not enough, because you need to use PCR to duplicate your DNA. So to duplicate that, there's three hairs. So make sure you just don't drop three hairs at the same time. Okay, you drop three hairs at the same time, maybe one day you will see your clone. Okay, it is actually, with the technology now, it is actually very easy to do that. It's just that due to ethical issues, we are not allowed to clone human or any organism. That's the only thing only. Yeah. Okay. So now, because of this, okay. So that's why the DNA needs to be tightly packed. It needs to be highly organized. So how is it going to highly organized? Basically, they use histone protein. Yeah, they will use histone protein. Okay. will be putting 8 histone protein together. So the 8 histone protein will form a cylinder. Okay, 8 histone protein will form a cylinder. Then the DNA will wrap around the cylinder. Okay, so basically 8 unit, 8 of this histone protein wrapped around by the DNA. This is considered as one basic unit of the DNA, of the chromatin. Okay, so this one basic unit is called what? It's called nucleosome. This one basic unit is called nucleosome. Okay, so basically the DNA wrapped around the histone protein to form nucleosome. So nucleosome will further condense each other, they will stack together, and then they become the chromatin. So when they become the chromatin is a rope, yeah? Then the chromatin, when you need to undergo the mitosis or meiosis, then the chromatin will further condense to become the chromosome. So that's how it works. And that's the structure. So yeah, that's what is shown here. Chromatin. This is chromatin, and this is one 8 histone protein, right? Then they form one nucleosome. Nucleosome coil become the chromatin. Then chromatin further condense and coil become the chromosome. Then next we'll look at the chromosome. Yeah, look at the chromosome, structure of the chromosome. So for the chromosome, right? Sometimes you will see a chromosome like this. Sometimes you will see a chromosome like this. Which one is called chromosome? Both also chromosome, yes. This one is named as chromosome. This one is also named as chromosome. Okay, so this one I call it as pride. This one is butterfly. Yeah, this one 2 wing only, so it's as pride. Okay, so this one is butterfly. Okay, so no matter it's a fly or butterfly, right? Both of these is called chromosome. The only what is the difference between them? The difference is that this is the chromosome before the replication. This is the chromosome after the replication. That is the only difference. Okay, so I think you all know replication. You learn about the cell cycle. In the cell cycle, there will be G1 phase, S phase, and G2 phase. Okay, and in the S phase, replication of the DNA occurs. This is what you have learned in the secondary school. In other words, this is one strand of the DNA. We have 46 strands of this DNA. Okay, so before our cell undergo S phase, during the G1 phase, for example, or when the cell is just finished dividing. Okay, so all the daughter cells or all the cells that haven't undergone the S phase, they will have 46 strands of this DNA. If let's say you let the 46 strands of the DNA condense, they will form a chromosome like this, like a fry. Okay, they will form a chromosome like this. Okay, and those cells, after undergoing S phase, after the S phase, replication occurs. So in replication, what actually happens? Replication is that they copy another strand of the DNA. So that will mean that the DNA is just not one strand. Every strand of the DNA, they copy it. So meaning that it's having 2 strands of the DNA. Okay? This one is double-stranded, this one is also double-stranded. Okay, it's directly copied. It's just that these 2 strands of the DNA are totally identical because the blue color one is totally copied, replicated by the DNA polymerase. This process we call replication. You will learn it in the next lecture. Right? So when this one, after the S phase, when you condense it to become the chromosome, you will form a chromosome like this. Because the chromosome will be 1 strand, 2 strands. The blue color one is actually the newly copied one. So you will have 46 strands of DNA after the S phase, after the replication. You will have 46 strands of DNA like this. Before, it will be like this. After, it will be like this. So that's why, no matter this one or this one, both of these are called chromosomes. Okay? But this one is only one only. Alright? So the whole thing is called chromosome. This one got 2. The whole thing we call it chromosome. But inside it, we know that this arm and this arm, we know that both of them are identical. Yeah, they are identical. Okay, so that's why we can call them as sister chromatid. Since this one and this one, they are the same. They are totally identical. We know it's copied from each other, right? So that's why we call it sister chromatid. Okay, sister chromatids. Okay, so that's why in mitosis, in meiosis, anaphase 1, anaphase 2, well, sometimes it is the chromosome being separated. Sometimes it is the sister chromatid being separated. Okay, so that's why you need to understand the concept first. Then later on in the phase, we will talk about what is being separated during the anaphase, or anaphase 1 or anaphase 2. Okay, but this is the concept that you need to know. Okay, so done. Then next, the chromosome, right? So of course there will be longer arm and shorter arm. Okay, so the shorter one we call it P arm. Longer one we call it Q arm. Sometimes it will be the upper part is longer, then the lower part is shorter. Sometimes it will be like this. Okay, so if let's say longer one, we don't care. We don't care whether it's up or down. As long as it's longer, it means it's Q arm. Shorter means P arm. Not necessarily the one that's on top is the shorter one, not necessarily. Sometimes it can be vice versa. Okay. So that would be the chromosome. Then the attachment point, not attachment point, yeah. So the center point. The center point of the chromosome, we call it centromere. We call it centromere. It's different from centrosome, yeah. So they're different meaning. Okay, so that's the structure of the chromosome. Okay, then next, telomere. What is telomere? Telomere is actually at the end cap of the arm of this chromosome. At the end cap of the arm of the chromosome, that is the telomere. Okay, why this telomere is important? Well, two. Yeah, basically you got two reasons. Okay, so first one is to preserve the DNA sequence, to preserve the gene sequence. Okay, it make sure that the gene sequence don't deteriorate. That's the first function. Then second function is that it prevents the chromosome to fuse together. The chromosome and another chromosome they fuse together to prevent that. So that is the two reasons why we need the telomere. So how come the telomeres can actually protect our gene, and why we need that? Okay. Well, the reason is very simple. Just now we talk about the replication. We talk about replication just now. Okay. So replication, well, in replication we will use the enzyme called DNA polymerase. Okay. So let's say this is the sequence. Supposed to be double-stranded, yeah. So it's just that I'm lazy and draw one. Okay. Supposed to be double-stranded, yeah. It should have the complementary base pairing. A pair with T, A pair with T, right? Supposed to be like this. But I only draw one. Okay. So it should be two strands. So in replication process, in the replication, DNA polymerase will bind. They will bind, and then they will add. They will add the sequence. Add. So they will produce a two strand. So another strand, it will also have another DNA polymerase. It will also add. So it will produce one double-stranded here, then another double-stranded here. So in other words, in the end, you will have two double-stranded DNA. So that's how the replication occurs. Okay, and that's how you have this. You have two double-stranded. So the red color line is one double-stranded DNA. The blue color line is another double-stranded DNA that's being copied. Okay, so let's say this is a sequence. So we know that on the DNA there will be gene. So for example, the blue color one is a gene. Let's put for the gene. Okay. So now the problem is that when the DNA polymerase replicate the DNA, they will start here, they will stick, and then they will copy along. So when they copy along, right? So this strand they will copy this way, this strand they will copy this way. Okay. So they will copy. So when they copy, what will happen is that they copy until the end, then they will detach. few sequence will not be copied because the polymerase is quite big. So polymerase it will work until here. Okay, then it will detach. So when it detach, right? So that will mean that the last few DNA sequence will not be copied. Okay, so if let's say you don't have the end cap, if you don't have the end cap like this, you don't have the rubbish sequence at the end. That means that what will not be copied? The gene will not be copied entirely. So every time you do the replication, that means one round of mitosis, some of the gene will be lost. If some of the gene will be lost, then how are you going to start? If let's say this is just a normal gene, let's say for black color hair, that one is fine. Then you will have blonde color, okay, or white color. But if let's say this is insulin gene, then you will have problem. Okay, so that's why if let's say if let's say you don't have this cap, you don't have this the DNA sequence that is not coding for DNA at the end of both sides. Every end you need. Okay, if you don't have that, then basically one round of mitosis is going to destroy the DNA, destroy the gene. One, it disturbs the gene, means the organelle will be. Okay, so that's why it needs a sequence, and this long sequence at the end, of course, it will be much longer. Okay, so this long sequence at the end, when it condenses to become a chromosome, it will form the telomere. So this one, telomere. Okay, so that's another strand. It will become a telomere. It will become a telomere. Okay, so that's how the telomere occurs, and that's how it protects the gene. And that's how it protects the gene. Okay. And some of the since DNA polymerase, every time they can copy until the end. So that means that this sequence and this sequence will become shorter and shorter. Every round you do the mitosis, one round of the cell division, the telomere will be shorter. Next round it will be shorter again. Next round it will be shorter again. Because why? Because every round before you want to undergo mitosis or meiosis, it has to undergo replication S phase. G1 phase, S phase, G2 phase. Then mitosis. So after mitosis, the two daughter cells will undergo the same thing. G1 phase, S phase again. So it will become shorter and shorter. The telomere will be shorter and shorter. Okay? So until the point where the telomere is gone. If the telomere is gone, then that means that the cell can no longer undergo the mitosis. If the cell undergoes the mitosis, then the cell will be dead. Why? Because the newly produced cell will be affecting their gene. If affecting the gene, normally the cell is dead. The newly produced daughter cell, after they form the daughter cell will be dead because their genes are affected. Okay? And this is why we are having aging and death. Okay? Our body cell, let's say your neuron, let's say your hair cell. Well, it will keep, it can, when you are growing, for example, your bone cell, when you are growing, yes, it can undergo mitosis, increase the number of the cell. Okay? But then normally it's after you reach puberty. After you reach puberty or you stop growing, that is when all the cells have been lacking of the telomere. So your cell can no longer undergo mitosis to replace it. For example, your lung tissue, the lung cell, the alveolar cell. It can no longer undergo the mitosis. If let's say undergo mitosis, you will be dead. No point. Okay? And if let's say they don't undergo mitosis anymore, so any cell, let's say you smoke, what happens? You take weight, what happens? It kills. After it kills, because your cell can no longer do mitosis, so that's why no one to replace that. So slowly the tissue will be dead, more and more cells die, and that's why aging and dying occurs. Okay? But maybe you will say, so then how about the stem cell? Our liver cell, the cell in the cortical cell in the testis, let's say, or the bone marrow. So all of these are the cells, tissue-specific stem cells that can keep on undergo mitosis for your whole life. So how about this cell? Why they can do the mitosis for the whole life? They can keep on undergo the cell division. Well, that is because these stem cells, they are having an enzyme, and the enzyme is called telomerase. Yeah, telomerase. They are having an enzyme. So these cells, they are having an enzyme, telomerase, that can keep on adding So every time after the mitosis, they can add it on the telomerase enzyme will add it on the telomere. So their telomere won't wear out. So with that, they can keep on doing the mitosis, okay, to replace those wear out cells, okay, to produce the new cell for the whole life. Okay, but it is only certain tissue-specific stem cell. Okay, only certain. So that's why human beings, they are still facing aging and age. Okay, so this finding is actually very important because this can be one of the a very hot topic now actually, right? Because a lot of scientists is actually finding the ways to achieve immortality. Yeah, so if let's say you want to live longer, you want to be immortal, this is actually the way. You need to find a way that can add the telomere for every single cell in you. If let's say you can do that, you can find that, then that means that yes, technically it's no longer winning Nobel Prize. It's like become immortal. Okay, it is. The scientist is actually trying to find that how to make other cells having the telomerase and having the endless telomere. If you can do that, that means that every single cell, any single cell in your body can actually regenerate. Okay, so yeah, if you are interested, maybe this can be one of your study topic for your PhD. Okay, then one day maybe you will find a way. Who knows, right? Okay, so this is what I explained just now. Yeah, when it is one strand of DNA, this is before the replication. It will be a fry like this. Okay, but after the replication, it will contain two double-stranded DNA. Yeah, two double-stranded DNA. Yeah, so this one means that it's going to form another fry. Okay, then after the mitosis, because during the mitosis or meiosis also, during the anaphase, the sister chromatid is going to be separated. So when sister chromatid being separated, so that's why the daughter cell will only having one strand of the DNA. Right? That is that having one chromosome like this. Okay, so this is before the S phase. This is after the S phase. This is after the anaphase. Okay, so don't worry. As for now, maybe some of you will have a clear picture of that. At least some of you maybe still learn that. It's fine. Okay, because this totally depends on what is your understanding during the secondary school. Okay, but it's fine. So if let's say you miss that, then it's okay. Later on, later on when we talk about chromosome behavior, right? From one phase to another phase, we will look at that. Okay, then next, well, being human, we are having 23 pairs of chromosomes. Why is it pairs of chromosomes? Why is it 23 pairs? It is because, well, we will receive one set of chromosomes from chromosome number 1 until chromosome 2, then plus an X or Y. You receive one chromosome 1 to chromosome 22 plus an X chromosome from your mother, which is from ovum. Okay? And then another set of chromosome number 1 until chromosome 22 plus a Y chromosome, okay, or X chromosome from your father, from the sperm. Fertilization occurs from the zygote and it becomes you. Okay, so that's why you have one set from father, one set from mother. Okay, so that's why we have 23 pairs. Yeah, we have 23 pairs of chromosomes. So total 46 chromosomes. So every single cell in us are having 46 chromosomes. So all of these type of cells we call it diploid cell, meaning that having two sets of chromosomes, two ends, diploid. Only when you want to produce the ovum or you want to produce the sperm cell, it undergoes meiosis, then the diploid cell will become haploid cell. That means that in the sperm or the ovum, it will contain half of this only. So for example, for chromosome number 1, when you produce the sperm, only either one will go into the sperm only. Chromosome number 2, only one. Chromosome number 3, only one. Chromosome number 4, only one. That means that only one set only. You won't have two. If you got two, your offspring is going to have mutation. Down syndrome, let's say. That means that if let's say in the sperm or in the ovum, got 2 chromosome 21. The rest is all 1, If let's say suddenly you have chromosome 21, that is 2. Then let's say your wife is normal. But normally Down syndrome is due to ovum. It's normally due to ovum. Okay? So if let's say in the ovum got 221, in the sperm is normal. You combine, it will having 321. 321, that is trisomy, that is the Down syndrome. That's how the mutation occurs. Okay, so that is that's why in mitosis or meiosis you have to ensure that our body have to ensure that it only have one set of chromosomes. It's important. Okay. All right. So now, just now I named chromosome 1 to chromosome 22. Chromosome number 1 to chromosome 22. Okay. So these are the chromosomes that contain the gene that code for your survival, that code for your metabolism, that code for your functioning of the organ. Okay, it's not related to sex determination. So this type of the chromosome we call it autosome. Yeah, we call it autosome. Okay, and we have a pair of the chromosome which is XY chromosome. Either girls will have XX and the male will have XY chromosome. So this XX or XY chromosome, this special pair of the chromosome, this is the chromosome 23. Yeah? This special pair of the chromosome is containing the gene for sex determination. It will determine whether you are a boy or you are a girl. All right? So this one we call it sex chromosome. Yeah, we call it sex chromosome. So that's the difference. Okay, but in the separation during the mitosis or meiosis, everything will be separated into half. You will only get one X or one Y only. Okay, in the sperm. Okay, or you for girls you will only produce one X. It cannot be two X. Again, if let's say something wrong, you got two X in the ovum or you got one X, one Y in the sperm, you fertilize with a normal gamete. Again, it will lead to either Klinefelter syndrome. It will lead to for example, the super male syndrome. All of this. Okay, so this one you will learn in Bio 3. Yeah. Okay, so just now I said a pair of chromosomes. Okay, so a pair of chromosomes means that you get one from the father, one from the mother. Why we say they are pair? Because they contain the same gene. Yeah, they contain the same gene. Okay, so they contain, for example, this is one pair of the chromosome. Let's say this is chromosome number 1. Yeah, chromosome pair number 1. Yeah, so chromosome pair number 1. Well, why we say they are the pair? You get one from father, one from mother. Why we say they are the pair? Because on the chromosome itself, it's actually having the same gene. For example, on this chromosome, let's say chromosome number 1. Well, this chromosome is having the gene that code for eye color. So that means that this one also having the gene that code for the eye color. Okay. Then if let's say over here got the gene that code for hair color, that means that this one also have the gene for hair color. Okay. It's just that it might be different version. In other words, different allele. So this one is coding for eye color. So it's just that maybe from your father one, your father, let's say it's black eye. Your mother one maybe is blue eye. So it's recessive. So you will have dominant or recessive. That determine different color. But then overall the gene is still coding for eye color. That means that it's having the same gene, it's just that maybe different allele. Okay? So hair color, maybe your father one is black color, your mother one maybe is blonde. Okay? So just dominant or recessive. But on the pair of the chromosome, it is having the same gene, same type of gene. It's just a different version and a different allele only. Okay? So that's why they are considered as one pair. So this pair, we will name it as homologous chromosome. We will name it as homologous chromosome. This is important because in the prophase one, it is the homologous chromosome that will pair up. Yeah. Okay, so that is the homologous chromosome. Yeah. All right. So for homologous chromosome, well, this is how we determine whether it's diploid or haploid. If let's say haploid, haploid means that in that, right? For example, human beings have 23 pairs of chromosomes. Okay. So in the haploid cell, it only contains 23 chromosomes. It don't have any homologous chromosome pair. It won't have any homologous chromosome pair in a haploid cell. But in a diploid cell means that every single chromosome will have a homologous chromosome pair. That's diploid. Okay, so that's the difference between N cell and 2N cell. That's the difference. Okay. Done. Then next, we'll look at the binary fission. Binary fission, this one is a simple one that's done by the prokaryotic cell, by prokaryotic cell. So you can see here the cell, right? The bacteria, for example, they can actually suddenly just elongate, pinch to become two cells, just like this. Very simple, very fast. Okay, and this process is the so-called binary fission. Binary fission is considered asexual because the genetic material in the newly produced cell will be totally identical. Exactly identical, exactly the same like the parent cell. It won't change. Yeah, it won't change at all. Okay, so let's look at the process of the binary fission. It is just a three-step process. Only three steps. Very simple, only three steps only. Okay, so the first step is they replicate their DNA. Their DNA here is the nuclear. Here is the nuclear, yeah. It's not the plasmid. Plasmid, they don't do like this. Okay, so it's their nuclear. So their nuclear will just replicate. That's it. One becomes two. That's it. It's circular DNA. So one becomes two. And it is not condensed with the sister protein to become any chromosome. No, it's not a chromosome at all. Right? So it's just DNA. So for that, they just directly replicate. That's the first step. Step number one, replication. Okay, then step number two. Step number two is elongation. So after they replicate, step number two is that you can see the cell will start to elongate. And the cell will start to elongate. So that is step number two. So when the cell elongates, so the DNA will move to different sites. Yeah, the DNA will move to different sites. That's it. That's the second step. Cell elongate, DNA move to different sites. Okay. Then step number three. Step number three is they will divide. Division. Right? Division. So after they elongate, after the DNA move to different sites, then next is that the plasma membrane will start to pinch in. The plasma membrane will start to pinch in. So it will pinch in until it pinch the whole cell into two. That is the third step. That's it. Very simple. So binary fission, that's why it can complete in 20 minutes. Okay, but for mitosis meiosis, it takes like 20 hours to 26 hours. Okay, so that's the difference. Okay, then next we'll move on to the cell cycle already. Yeah, that is for eukaryotic cell. This one is eukaryotic cell, yeah. Okay, so eukaryotic cell because the presence of the nucleus, the presence of the membrane-bound organelle, and also the presence of multiple chromosomes. Because of these three reasons, now the eukaryotic cell need to undergo a more complex process in order to separate, in order to divide the cell. Okay, this is mitosis or meiosis. Okay, so before going through that, yeah, so there are terms that we need to highlight here. Okay, this is the common terms that some students will confuse. So first one, centromere versus the kinetochore. And then one more is centromere versus centrosome. And this is the common words that the student will write wrongly. Okay? So some students, they will confuse it. They thought it is centromere, but they go and write kinetochore. Or some students, it is a kinetochore, but they go and write centromere. Okay? So this is a common careless mistake. Yeah? So what is a centromere? Centromere is actually on the chromosome itself. Okay, centromere is the center part here. It's the center part here of the chromosome. Okay, it's the attachment point where the two chromatids are joined together. So that is the centromere. Okay, even a fly one also got centromere. This one is a centromere. Butterfly also got centromere. This is the centromere. Okay, it's the center point of the chromosome. Right, so that's the centromere. So kinetochore Kinetochore is actually the protein that attached to the centromere. So that is a kinetochore, the yellow color one. So kinetochore is the protein that attached to the centromere. So why they need the kinetochore? Because kinetochore is the protein that attached to the centromere, so that they allow the spindle fiber or the microtubule to bind to the kinetochore. Okay, so the spindle fiber or the microtubule, they will bind to the kinetochore, then kinetochore bind to the centromere. So that's why they can later pull, right? Pull the chromosome. Okay, so that's how. That's a difference. Then another terms that normally confuse centromere and centrosome. Centrosome, different thing. Okay, centrosome is actually a pair of centriole. It's actually a pair of centriole. So a pair of centriole, they are aligning at 90 degrees perpendicular to each other. So that one is the so-called centrosome. One centrosome is a pair of centriole. Okay. So before the G2 phase, one cell contains only one centrosome. But then during the G2 phase, before the mitosis or meiosis, right, the centrosome will be duplicated. So now, after the G2 phase, that means that when they undergoing the prophase, right, the cell, every cell will have one pair of centrosomes. Every cell will have two centrosomes. Okay, so centrosome is this one, yeah, is actually a pair of centriole. So centrosome and centromere are different things. Those are wrong things. Right? So for the centrosome, well, we know that it is made of centriole, and we know that centriole is made of microtubule. Yeah, it's made of microtubule. So that means what? That means that they are able to work as MTOC. Yeah, the MTOC. So that's why, yep, every single centriole, yeah, they can actually produce so many microtubule. And that is actually the spindle fiber. Okay, they can elongate. The microtubule that make the centriole, they can elongate and form the microtubule, form the spindle fiber. Okay, so yep, it will be having a pair of centrosomes. So that's why the centrosome, yeah, when during the prophase, during the metaphase, you will move to different poles. Yeah, you will move to different poles. Then in the end, you'll pull the chromosome to different poles. All right, so that is the centrosome. So they can form so many so many spindle fiber. So the spindle fiber is long and go and bind to the chromosome because spindle fiber. Or those spindle fiber that actually cause the elongation of the cell, that one because spindle fiber. But those that are very short one, we call ester. Okay, not because they can produce so many spindle fiber, not every spindle fiber are needed. Okay, so that's why those that are short one we call ester. Okay, then now we look at the cell cycle. Now you know the all the terms already. I will confuse that. Then now we look at the cell cycle. Yeah. So cell cycle there will be G1 phase, S phase, G2 phase, and then the M phase. M phase it can be mitosis or meiosis. The M phase can be mitosis or meiosis. Right? Either one. Because either it is mitosis or meiosis, so it has to undergo G1, S, G2, and then M phase. So you need to know what happened in the G1, S, and G2 phase. Okay? So G1 phase, S phase, G2 phase, all these three phases, G1, S, G2 phase, collectively we call them as interphase. Yeah, interphase. This is the interphase. G1, S, and G2, that is the interphase. Interphase, right, it takes a lot of time. The whole cell cycle, right, it takes around 80 to 90% of the time. So the mitosis, right, or meiosis, right, it only takes like 10 to 20% of the time. Okay. So what happened in G1? What happened in S? What happened in G2? G1 phase is the so-called growth 1. Growth phase 1. That's the G1 phase. Okay? G1, yeah? G1 is a growth phase 1. Growth phase 1 is basically after, right after the mitosis. That means that after the cell divide, one cell undergo mitosis will become two daughter cells. So the two daughter cells that's newly produced, right, they are very small, very small. Okay. So what they will do is that they will undergo the G1 phase. So in the G1 phase, what they do is they grow. Yeah, they grow. So in other words, what is actively done here? First one, synthesize the protein. They will synthesize all those proteins that's needed for metabolism, that's needed for growing. So in other words, they will increase in their cytoplasm. Yeah, they will increase in their cytoplasm. They will grow in the size. So that's what happened in G1 phase. So they will also produce some organelle. So those organelle will be the organelle that's important for the metabolism or for the growth or the function of the cell. Example, ribosome. Example, Golgi apparatus. Example, the RAF-ER. They will produce this type of organelle. Okay, they will produce this type of organelle. Okay. Then after that, the cell grow to certain size already. After they grow to certain size already, then they will undergo the S phase. They will go into the S phase. So during the S phase, only one thing happens. It is replication of the DNA. This is when the fry becomes butterfly. One strand of DNA becomes two strands of DNA. Okay, one double strand becomes two double strands. That is the replication. That's where replication occurs. Okay, that's S phase. The only thing happened only. Yeah. Then next, G2 phase. G2 phase is growth phase 2. Growth phase 2, well, during this time, well, it's almost the same. Synthesize the protein, duplicate the organelle, produce the organelle, duplicate the organelle. This one also duplicate organelle. So this one also duplicate organelle. This one synthesize the protein. This one also synthesize the protein. Okay, but what is the difference? The protein, the organelle that's being synthesized here will be the protein and organelle that's involved in cell metabolism and cell function, cell growth. In G2 phase, the protein that's being produced, for example, will be like tubulin protein. This is a protein that later will be used to form the spindle fiber, to form the microtubule. Okay? Organelle that's being duplicated here, what will be the organelle? The organelle will be, for example, centrosome. The centrosome, all the cells should have one only, right? Okay, but during the G2 phase, the centrosome will become two. So later on, the centrosome will move to different poles. So that later on, the centrosome will separate into two cells. So they are actually double the organelle that is needed normally for cell division. They produce the protein that's needed for cell division. So that is the difference between G1 phase and G2 phase. Because if you do extra reading or you sometimes look at the reference book or in the internet resources, they will tell you, oh, G1 phase produce the protein, they duplicate the organelle, G2 phase also do the same thing. So some students they were confused. If they are the same thing, then what is the difference? Okay, they are different because the purpose they produce the protein and the organelle is different. Okay, so then G2 phase. So after G2 phase done, then only then they will go into the M phase. Yeah, then M phase is mitosis or meiosis. Okay, then they produce the daughter cell, then they continue. So the new daughter cell will undergo G1 and G2, then only then they can replicate again. Okay, so that is how it occurs. So that is the cell cycle. So in the cell cycle, there's one more important thing we call checkpoint. Checkpoint in the cell cycle. Okay, so you have learned about the apoptosis before this, right? Apoptosis. Okay, so apoptosis will occur when? Well, actually, after G1 phase, there will be a checkpoint. After S phase, there will be a checkpoint. After G2 phase, there will be a checkpoint. They will actually check. And they will check themselves. Okay, so if let's say after G1 phase, they found out that they are different from their peers or something wrong with them, then they will undergo apoptosis. If after S phase, they will check their DNA. If their DNA is different, then they will undergo apoptosis. If after G2 phase, right, they found out that, let's say, centrosome, they didn't replicate the centrosome. Let's say the cell only contains one centrosome, then the cell will undergo apoptosis. Okay, so normally this checkpoint it will cause apoptosis, and most of the time this apoptosis occurs after S phase or G2 phase. G1 phase normally no. G1 phase is very lesser, okay, but compared to S phase and G2 phase, this is where the after this interface. After the interface going through the checkpoint, then only then the cell going through the M phase. That is the mitosis or meiosis. Okay? Then after the mitosis and meiosis, well, mitosis or meiosis is actually the division of the nucleus. So after the division of nucleus, you need to divide the cytoplasm. So that phase is called cytokinesis. But cytokinesis, first one is very short. Second one is because it normally occurs together with the telophase. And then the halfway of the telophase, it starts cytokinesis. Okay, so that's why it's actually very short. It's very short. Okay, then after that, you're done. That will produce the new cell. Okay, so if let's say the cell want to keep dividing, then they will stay inside the cell cycle. They will stay inside. Okay, so all your stem cell. Okay, all the stem cells in the testis, in the bone marrow, the skin cell, the liver cell, all of that, they are still staying inside the cell cycle. So that's why they can keep on undergo mitosis or meiosis. Okay, but other cell, other cell, for example, your neuron cell, for example, your lung cell, okay, all the other muscle cell, all of these cells, they stop replicating. I mean, they stop dividing. So all of these cells, right, after they produce one become two, then after that, it will exit the cell cycle. So when they exit the cell cycle, this will be the cell undergoing GO phase. Right? G0, yeah, so GO phase. So GO phase will mean that this is mitotic dormancy. These type of cells that go into the GO phase will no longer undergo mitosis because they quit the cell cycle. So after this, the cell will undergo specialization and differentiation. So it becomes a specific tissue, and this specific tissue, since they are no longer able to do any mitosis, they quit the cell cycle already. So that's why it means that if let's say it's dead, it's dead. If it's dead, it means you lost the cell. You don't have that anymore. So that's why your neuron, the lung, so all of this, that's the reason. Okay, so that is the GO phase. Yeah. But of course, there are some cells that actually can enter GO phase and then go back into cell cycle. For example, the stem cell in ovary, in female ovary. So in female ovary, when they are in fetus, they are still inside the cell cycle. Once they are born, when they become baby, then the stem cell inside the ovary will change to become GO phase. Then when after 12 years, after reach the puberty, due to the high level of estrogen and progesterone, then only then, FSH, then only then it re-enter the cell cycle. So there are cells that can actually exit first, stay dormant, and then re-enter the cell cycle. Okay? But most of our cells cannot, because most of our cells, once they exit, then they will specialize. Once they specialize, they won't be able to undergo the mitosis anymore. Okay, so that's the difference. Yeah? That's the cell cycle. So once we have learned this, then now we can start to look at the phase, right? Step for mitosis and also meiosis. Okay, so first one, interface. Yeah, interface. So after the interface, well, interface it can be G1, it can be G2, it can be S phase. To you, under the microscope, they look almost the same. Exactly the same. No difference. So the cell under the interface just looks like a normal cell. Okay? Because why? Because the chromosome, the chromatin does not condense yet. The nucleus is still intact. So that's why when under the microscope you see nothing, you only see nucleus and cell, normal cell. It's just like normal cell. You won't be able to differentiate a cell in the interface or a cell that is already exit the cell cycle. It's the same, it looks the same. Because nucleus is still intact, you won't see anything inside. Chromatin also you cannot see also. Okay? So that's why you only see the nucleus. So that is the interface. Alright? So then after that, yes, we will look at the mitosis. Okay? So no matter what phase is that, what we are going to discuss. 4 things. It's the 4 major things. Okay, that is very important for your understanding. Okay, so once you know this, then yes, should be fine. Right? So the first thing is in every phase, we will look at their chromosome behavior. That's the first thing. nucleus behavior. That's the second thing. Third thing is the spindle fiber behavior. This is the three things that we will look at in the face. Okay. Then one last thing is the microscopic view. Because it is very common that we will give you a micrograph. We give you a picture, micrograph, under the microscope, how it looks like, the cell looks like. So you have to identify this one is interface, prophase, metaphase, prometaphase, anaphase, or telophase. You have to figure it out. Okay, you have to figure it out. By looking at the picture, you need to know that. Okay, so this is a very common thing that it's asked about the mitosis and meiosis. You must be able to identify the phase based on the microgram. Alright, so that is the four things that we are going to discuss one by one. So when we go through the phase, we will talk about the first few things first. So for the microgram, I will leave it to the last after we finish all four phases in, and then only we talk about the microgram. Okay. So for microgram, well, actually very simple. You don't worry. Okay. Because based on my experience, I have actually summarized the tips for you to actually identify it. You just have to, when you see the picture, there's only a few things that you need to look at. When you look at that two things, automatically, for sure, you are able to differentiate it. Okay. So don't worry too much. Because microgram is totally new to you, okay? Because you are not having the experience. So that's why a lot of students get confused and they are not able to identify it. But don't worry, you can just use my tips. You just look at the 2 points. Every picture you look at 2 points, you know about this battery. Okay? So later we'll talk about that. Alright, so now mitosis. Yeah, mitosis. So for the mitosis, before we start, you can draw the table together with me. Okay, so draw a table like this. Alright, so for mitosis, you need 1, 2, 3, 4 columns, and then 6 rows. 1, 2, 3, 4, 5, 6. That's 6 rows. No need to copy first, you just draw the box first. Then later, when I go through, then you do down at the same time. And later we do it together. This column you make it smaller because when I draw, I didn't, I leave it too big already, so that's why not enough space for here and here. Okay, so this column you make it smaller, this one bigger column, this one smaller column, this one bigger column. Then for the row, you don't want to draw the line also now, right? Later, after you finish, only you draw the line. Okay, to draw a table, but then as we go through the different phase, then I will tell what is the important thing and only you do now. Okay, then it will be. And then you don't need to write perfectly or nicely. Just simply draw. Okay, because after that you need to do revision. So when you do the revision, you draw the table properly. And then during that time only you write properly. You go back, then it will be a revision. Then after you finish the table, if let's say you can finish the table without referring to anything and then you can explain to yourself, that means that you are done with this all mitosis and meiosis. You understand the whole thing. Okay, and later I will mention about the key also. What are the key? Because sometimes they will ask you to compare, for example, they will ask you to compare the anaphase 1 and anaphase 2. Sometimes they will ask you to compare prophase and prophase 1. Sometimes they will ask you to compare prophase 1 and prophase 2. So all of these things you need to know what is the difference and what is the similar thing. Because there are so many similar things. Okay? Only sometimes it's just more different to it, but the thing is whether you understand it or whether you can detect the differences or not. That's the key. Okay? So to make it simple for you, to summarize all of this for you and prevent the confusion, so I will point that out. And then later you can just highlight. Okay. You don't need copy first, you just write down prophase. If you want to split the table also can. If you want to split mitosis table and meiosis table also can. But for me, I tend to combine everything because it's easier for you to refer when you want to compare everything. Because it's not just comparing prophase 1 and prophase 2 only. It will be comparing mitosis and meiosis also. Right? So it's better that you combine. But if let's say you want to separate it, also can. It depends on you. So as for now, you just simply drop that. Yeah, just simply drop. No need to write beautifully. That one you leave it. You do it at home. Okay, yeah, ready? Everyone ready? All the line, you just draw the line and table. Okay, then on top, chromosome. This is chromosome behavior, nucleus behavior, spindle fiber behavior. Okay, so this is the three things that we will look at. And you know, the questions are level 1 until level 4 question. Normally, this is, especially level 3 and level 4 question where they test your understanding one. They are testing on these three things only. Okay, so we will look at it one by one. Yeah. Okay, let's go. So now, four phase of the mitosis are prophase, metaphase, anaphase, and telophase, right? But the prophase is very, very long. In the whole mitosis, prophase can take up to 80% of the time. Right? It can take up to 80%. So that means that most of the time the cell is under prophase. Okay? And if let's say you take for example, if you want to observe the cell under this phase, right? Normally you will take those actively dividing part. For example, we will take the onion root. Onion root, right? So onion root, that part is actually having a lot of mitosis occurring at the same time. Okay? If you observe under the microscope, you know what? Most of the cell is actually under the interface of prophase. You will see most of the cell. Very rare you can find the cell that's under metaphase or anaphase or telophase. Because why? Because they are too short. The phase are very fast. So that's why most of the cell is under prophase. So that means what? That means that yes, prophase is actually taking a long, long time. Okay? 80% of the time is actually under prophase. So because it's so long, so that's why we will further divide them into the early prophase and also the late prophase. So the late prophase, we can also call it as prometaphase. Okay, prophase, you can separate them, further categorize them based on what happened. Yeah, based on what happened. Yeah. Okay, we can further categorize them as early prophase and late prophase. Okay, so the mid to late prophase, mid to late prophase is normally called prometaphase. For us, I accept both. You can write mid to late prophase also can. You want to write prometaphase also can. Okay, it's the same thing, because sometimes if you refer to the reference book or the external resources, they will use the term prometaphase instead of late prophase, but it's actually the same thing. Okay, so we will look at it one by one. Yeah, we'll look at it one by one. Okay. For mitosis is one cell to become two cells. Okay, and in mitosis is one diploid cell to become two diploid cells. The daughter cell will be totally genetically identical. Right? So we look at the phase now. So first one, early prophase. So after the interphase, then now it becomes the early prophase. So once it enters the prophase, what happened? Yeah, what happened? First one, we look at the chromosome behavior. Okay. In the interphase, the chromosome is the DNA still exists as chromatin. Once they entering the early prophase, so what will happen is that chromatin will start to condense to become the chromosome. And this one you can spot it in the microscope. Yeah? So later I will talk about this. Okay, so in the chromosome behavior, the chromatin started to condense to form the chromosome. Later I will link it to the microscopic picture. Okay, so now I just talk about these 3%. Okay, so that's the chromosome behavior. They start to condense. Chromatin condense to become the chromosome. The terms coil, condense, decondense, uncoil. This one, please remember, I don't use it wrongly because in 125, so many of them write wrongly in the DOS, and then they lost the 3 marks. That's a lot. Okay. Coil, condense will mean that from chromatin become chromosome. Okay. from chromosome become chromatin, it's called decondense or uncoiled. A lot of the students in 125 they write oppositely. Okay, so please remember, yeah. So that is the chromosome behavior. Second, we talk about nucleus behavior. Nucleus behavior, what happened here? Nucleus behavior is the nuclear membrane will start to fragment. The nuclear membrane will break into fragments also, or you can say start to fragment. Meaning what? Meaning that the nuclear membrane is still there. It is still there. It's just that they fragment or they become pieces. They become the pieces. Okay. Next, spindle fiber behavior. So spindle fiber behavior, spindle fiber is just being produced now. It's just started to elongate. Started to be produced or started to be formed. So that's the spindle fiber behavior. Okay, so this is the tree behavior. So we look at the so you can see there. So chromosome behavior, chromatin start to condense to become chromosome. In other words, you only started to be visible. Only started to be visible. Okay. Then nuclear, the nucleus behavior, nuclear membrane started to fragment. They are still there. They are still there. Okay. So normally most of the cells under the microscope, whenever the nuclear membrane starts to fragment, normally the nucleoli or nucleus disappear. Normally disappear. But sometimes they will still appear. But this is not the key point to identify the phase. Okay? But nucleus behavior is like this. Okay? Then spindle fiber behavior, the only start forming, not yet attached to the chromosome. Just started to forming. So that's why you can see the centrosome just started to move to the nucleus. They are still close to each other. They just started to move. I haven't reached the nucleus. Okay, so for mitosis, I will give you more time. Okay, later the meiosis very simple. Meiosis, a lot of it is just copy only because it's the same. A lot of the phase is the same actually. Okay. Then next, prometaphase, which is a late prophase. Prometaphase, late prophase. What happened during this time? First thing, chromosome behavior. So during this time, all the chromatin has fully condensed to become the chromosome. It has become the chromosome. During the prometaphase, it has become the chromosome. Okay? Next. Nucleus behavior. During this time, the nuclear membrane has disintegrated and most of it disappeared. Okay? Most of the nuclear membrane disintegrate and disappear. So that's why under the microscope you won't see the shape. You will see it start to spread out. Okay, this one you will discuss later under the microscope. Okay, spindle fiber behavior. Spindle fiber behavior, well, the spindle fiber has been formed and then some of it has started to attach to the chromosome. Some of it has started to attach to the chromosome. So that's the spindle fiber behavior. Okay, so my map. Okay, so it has now become chromosomatic. It's no longer chromatin. Over here, you still can see chromatin. This one no more. Okay, it has fully condensed. Okay, so it's very visible now, right? So what about nucleus? Disappeared. No more. Disintegrate and disappear. Okay, then spindle fiber, they have actually fully formed, then attached to the chromosome. That is the prometaphase. Yeah. Sorry? Yes. Yes. Yeah, you can see it. You can say that the nucleus disappeared. Okay. Then next, we'll look at the metaphase. So in metaphase, what happened? In the metaphase chromosome behavior, all the chromosomes will now be aligned at the metaphase plate. Chromosome will be aligned at the metaphase plate. Okay? Nucleus behavior disappears. There's no nucleus. Spindle fiber behavior. Spindle fiber behavior. Well, it is the spindle fiber that fully attached to all the chromosomes. It is the microtubule or the spindle fiber that push all these chromosomes and align them in the center. It is the spindle fiber. So in other words, during this time, this is where the spindle fiber is the longest. And this is when it's longest. Compared to other phases, this is when it's longest. Okay, so in the table, chromosome, they are all aligned. It is named as chromosome now. You cannot say it's chromatid, or you cannot say it's a sister chromatid, or you cannot say it's a chromatid. No, it's wrong. It's chromosome. Chromosomes are aligned at the metaphase plate. Okay, then nucleus disappeared, and then the spindle fiber. This is where it's going to be developed because they are the one that actually push the chromosome and align them to the center, the metaphase plate. So this is basically the longest time, and when the spindle fiber is the longest. And this is important because for those like, for example, level 4 questions. Level 4 questions, sometimes they will give you a graph. So in that graph, they will tell you that this is the length of the spindle fiber. So you need to know that if, let's say, they are showing you like this, a graph like this. Let's say they are showing you a graph like this, okay? Then, if let's say they tell you that the spindle fiber is like this and this. This is the length of spindle fiber. So you need to know that this point is what? This point is the metaphase. Okay, this is how the analysis level 4 question can ask. Okay, then how about this point? Why it started from 0? Or sometimes they will ask you whether the graph will start from 0 or not. The answer is yes, that the graph should start from 0. Because why? Because during interface, spindle fiber will not be formed. Okay, so that's why all of these things. This is basically when it brings the question up to a higher order question, the level 4 question. Okay, so that's why spindle fiber behavior is important also. And that's why we talk about that. When it is longer, when it is. And metaphase is when it's longer. So you see the peak. Yeah. Okay, then next we look at anaphase. Yeah, we look at anaphase. Okay, so for anaphase, chromosome behavior. Well, during this time, anaphase will be in the anaphase chromosome will be separated. Okay, chromosome will be separated. Alright. So, but the thing is that what is the specific terms? Because in secondary school, in mitosis, they will say chromosomes are being separated. In meiosis 1, they will say the homologous chromosome being separated. In meiosis 2, sister chromatid being separated. Alright? But when you say mitosis chromosome being separated, is it actually specific enough? Actually, it's not. Okay? Why? Because this is a chromosome. This is also a chromosome. Right? So, when you say chromosomes are being separated, it's a bit confusing actually. Okay? So, since we know that what is being aligned at the metaphase is a butterfly. So that means that this is being aligned at metaphase. So during the anaphase, it is this one being separated. It is this one going here, this one going another pole. So in other words, this is actually sister chromatids. So a lot of students, they don't understand how come in some of the books they write sister chromatids, some of the books they write chromosome being separated. Because this one, whole thing is called chromosome. After being separated, this one is still called chromosome. It's just that this will create confusion. Okay, especially when you compare with the meiosis 2. So that's why, to make it simple, just remember, over here, it is the sister chromatid being separated. So that's the chromosome behavior. Yes, sister chromatid being separated. Nucleus disappeared. There's still no nucleus. Okay. Then spindle fiber behavior. Spindle fiber, They shorten. Spindle fiber, they shorten. They shorten in order to pull the chromosome to different poles. It's due to spindle fiber. That's the shortening of the spindle fiber. Okay. So talking about the behavior of the spindle fiber, that's one more thing. Because during this time, during the anaphase, when they pull the sister chromatid to different poles, the cell will actually elongate. So when the cell elongate, how come the cell will elongate? The cell elongate because of what? Because the spindle fiber, also spindle fiber. But the thing is, what is different is that those spindle fibers that connected to the chromosome, to the sister chromatid, okay, they will shorten. They will shorten in order to pull it to different poles. But the elongation of the cell would be due to the spindle fiber that's not connected to the chromosome. So for those spindle fiber that's not connected to the chromosome, what they will do that they will elongate in order to cause the cell to elongate. They push the cell to elongate. Okay, so that's what is happening. Yeah, that's what you need to know. Yeah, so that's why in the summary, you can see here. For chromosome behavior, anaphase, sister chromatid will move to different poles. Okay. Nucleus disappeared. For spindle fiber behavior, shorten one is to pull the chromosome, pull the sister chromatid to different poles. Okay. Those that's not attaching to any of the sister chromatid, they will elongate. They elongate so that the cell elongates. Okay. So that is a two different things. But when it comes to analysis of the graph, like what I mentioned just now, let's say it's a graph like this. Okay. We only look at the length of the spindle fiber that is connected to the chromosome. Okay, we don't care about the spindle fiber that is causing the elongation of the cell. We don't care that. Okay, get it? So this is the only thing that you need to know. And in the anaphase, how the spindle fiber behavior will be. It depends on whether the spindle fiber connected or attached to the sister chromatid or not. Okay. Then last phase would be the telophase. Yeah, telophase. Okay, so for the telophase, well, in telophase is when the sister chromatid reach the opposite pole. And so chromosome behavior, once they reach the opposite pole, then these sister chromatid will start to decondense. They will start to uncoil back to the chromatid. And they will start to uncoil. They will start to decondense to become the chromatid. But during this time, we still call them as a sister chromatid. It's just that sister chromatid start to uncoil. Okay. Nucleus behavior, the nuclear membrane will start to reform. Nuclear membrane will start to reform, nuclei will start to reform. Nuclear cell will start to reform. How about spindle fiber? Spindle fiber, because they actually reach the opposite pole already, so this spindle fiber will start to disintegrate, they will start to disappear. Okay, so you can see here, in telophase, chromosomes start to uncoil, so that's why it starts to reforming the chromatid. So when they're reforming the chromatid, you can see it start to disappear. Okay, then in nucleus, nuclear membrane will reform, nucleus will reform, spindle fiber will start to disappear. Okay, so with that, that is the behavior of the chromosome, nucleus, and spindle fiber in the mitosis. So I give you 1 minute to complete your table.
is but it's not attached to chromosome. How can it attach?
Tetramer means what? Later we'll talk about.
But
It's a pair.
So it's different from chromosome. The way the classify it.
Which one is not attached to?
You see the spindle fiber and this attached to
during the starting. This is the early prophase. No. Tetrad is mid to late prophase. Okay, completed. Yeah, completed. Yeah. So we completed. Now we finish the mitosis first. Okay. So after the telophase, that would be what? That would be the completion to divide the nucleus. Okay, but cytoplasm not yet. Cytoplasm is the cytokinesis. Yeah, cytoplasm. You have to divide the cytoplasm, which is cytokinesis. Okay. So now the thing is that cytokinesis. This is one of the common mistakes. Is that a lot of students will thought that okay, finish telophase then only cytokinesis. No, yeah. Most of the time, cytokinesis will occur halfway during the telophase. Halfway of the telophase, yeah, when the chromosomes start to decondense, right? During halfway of the telophase, the cytokinesis will start already. Okay. So cytokinesis they will occur in two ways. It depends on what type of cell it has. Is it a plant cell or is it an animal cell? If let's say it's a plant cell, then they are going to use what? They are going to use the cleavage furrow, right, to do the cytokinesis, the cleavage furrow. If let's say it's a plant cell they have cell wall, so that's why they are going to use the cell plate. And they're going to use the cell plate. Okay. So how the process will be? Yeah, I will use the picture to describe that. Yeah. So that you can see that. Normally, those questions that ask for description on this cytokinesis, normally 1 or 2 marks. 1 mark or 2 marks. It won't be 3 or 4 marks. It's impossible. Okay? To describe the cytokinesis is basically 2 steps only. Either 1 or 2 steps only. So that's why maximum 2 points only. Yeah, maximum 2 points only. Okay? So how are you going to describe this? What is the keyword to describe the cytokinesis? If let's say it is using cleavage furrow, if let's say it's for animal cell, yeah? For animal cell, what are the 2 points? Well, the first one is you have to talk about the contractile ring. It is the contractile ring. Okay? So the contractile ring will contract. The contractile ring here, it will contract. It contracts to form the cleavage furrow. So this is how it looks like under the microscope. The contractile ring will contract to form the cleavage furrow. That is the first point. Okay. So when you form the cleavage furrow, second point, then these will pinch in. Right? It will pinch in and eventually pinch the cell to become 2 cells. That is the second point. They will pinch in and eventually separate the cell into 2 cells. Okay, so that is for cleavage furrow 2 points. Contractile ring, cleavage furrow, pinch in, separate the cell into 2. So how about the cell plate one? For plant cell. For plant cell, well, first one is always the cell will deposit the vesicle to the center. Okay, the cell will deposit the vesicle at the center of the cell. That's the first one. So after that, these vesicles will fuse together to form a cell plate. The vesicle will fuse together to form a cell plate, and eventually this cell plate will grow to become the new cell wall and separate the 2 cells. So first point, vesicle, deposition of the vesicle. Second point, formation of the cell plate and becoming the cell wall to separate. So that is the 2 points. Okay, so that is for cytokinesis. It depends on which one. Okay, so we have done with the three things, right? Chromosome behavior, nucleus behavior, and also the interphase behavior. Now, point number 4. Okay, the fourth thing. Fourth thing is, how are you going to identify based on the microscopy, based on the micrograph? What are the points that you look at? What are the key points that you look at the picture in order to identify? Okay, so now first thing first, interface. Interface, normally students will confuse the interface with the prophase. And normally this is interface, this is prophase. Okay, normally will be confused here. So how to differentiate them? How to differentiate this? What is the key point that we will look at? Okay, so first normally we look at based on the chromosome behavior and also the nucleus behavior. When you look at the picture, if let's say you can't see any big dot, you can't see any line, that means what? That means that the chromosome is not formed yet, it's still in chromatin. Second, the nucleus is still intact. If the nucleus is still intact, you will only see nucleus. You won't see any other thing. Because nucleus is still intact, the light microscope cannot penetrate. Light microscope, you can only see nucleus, you cannot see what is inside the nucleus. Okay, so that's why when you see a normal cell with a normal nucleus, that is interface. Okay, then how about the prophase? Prophase is when you start to see thin line. You will start to see the line. Okay, you will start to see the line. Okay, nucleus. Well, during this time, the nuclear membrane just started to fragment. Because it started to fragment, that means that there are holes. So that's why you can see the thin line. Because if the nucleus is still intact, you can't see, even they condense to become chromosome, also you can't see anything. Okay, but because in prophase, you know that the nuclear membrane has started to fragment, so that's why you can see. Okay, you will see thin line because why? Because the chromatin has started to form the chromosome. It started to condense to become the chromosome, so that's why you will see thin line. Not that clear, but then at least you can see the line. So that's the key. And because the nucleus is just started to fragment, so that's why the nucleus, the shape is still intact. You can still see the shape. Okay, you still can see the shape. Then next, a lot of students will also confuse between the prophase and the prometaphase. Prophase and prometaphase, how to differentiate them? Okay, based on chromosome behavior, in prometaphase, all the chromatin has now condensed to become a chromosome. So that's why the chromosome, the line will be thicker compared to the prophase. The line will be thicker, more obvious, darker. Okay, so that is the first point that you can see. Second point, you can look at the shape of the nucleus. Okay, you can look at the shape of the nucleus. Okay, the prophase one, the shape, right, is still like a normal nucleus shape. It's still like an oval shape. You can see that. And the shape is still very clear. Okay, why? Because the nuclear membrane just started to fragment. So that's why the shape is still there. Prometaphase, the nuclear membrane disappeared already. So that means that during this time, the nuclear membrane all fully disintegrated and disappeared. So that's why the chromosome actually spread out. So you will see that they spread out. Okay, they spread out, they are no longer in this shape. So that is the 2 points that always we look at in order to confirm what phase is that. Okay, then next, metaphase. Metaphase, a lot of students will confuse the metaphase with the prometaphase. Because in metaphase, right, the chromosome is fully condensed, right? So that's why it is also thick, dark chromosome, thick and dark line. Okay, so how to differentiate this one and this one? Because this is out of shape already, right? This is also out of shape. So how to actually differentiate it in the micrograph? First one, chromosome behavior. We know that during this time, the chromosomes are all aligned at the metaphase plate. That's the key. So that means what? That means that if you look at that, right, this one you won't see a line. This one you will see a line. But sometimes it's hard for you to see a line. So if let's say it's hard for you to see a line, then what is the next point that you can use? Same thing, still chromosome behavior. Because they are aligned at the metaphase plate, that means what? That means that these chromosomes, some will be long, some will be short. Chromosomes, some will be long, some will be short. Right? Okay, so that will mean that you look at the space in between that. You can say that, you can see that the space in between these, right? In between the chromosome is larger compared to this one. The space. Because, and then, some long, some short. So that is the space. So if you look at the shape, the space, so you will be able to identify that. Because when it's aligned at the metaphase plate, well, it will be space, or bigger space in between. So with that, you can confirm this is actually the metaphase. Either you see a line, okay, but this, for example, this picture is not that clear. You not really can see a line. Okay, then this one, you will use the space in between to identify. Then next, anaphase. Anaphase simple, Anaphase when it's being separated. Okay, some students they will confuse between anaphase and the telophase. So how to differentiate that? Okay, because telophase also sometimes it is like starting before the nucleus or not starting before the nucleus. For example, this one not yet. Okay, so some students will tell this is anaphase or telophase. So anaphase or telophase very simple, same thing, chromosome behavior. Okay, over here the sister chromatin is separated. Over here, sister chromatin has reached the pole, and then they started to decondense, they started to uncoil to become the chromatin. Okay, so that means that sometime over here it will be blurred. Starting some parts it will be blurred. And very obvious thing is what? This one will be clumped together. Okay, this one, all the chromosomes will be clumped together. This one, not yet. Not clumped. This one clumped together. So those that clumped together one, definitely telophase. And sometimes you can see a line in between. A cell plate line. If you can see the cell plate line, that's called telophase. Okay, so these are all the key points that you look at in order to identify, to make the identification. Okay, so with that, yes, after this, hopefully, this type of question will be an easy mark for you. Okay, you know the point, then you can differentiate it easily. Okay, then this will be free mark for you already. Okay, so I have give you some extra picture for you to see that. Okay, so this one prophase, you can see that it starts to have a dot. Okay, it starts to have a dot. Sometimes nucleus will be a bit confusing. So normally I don't look at nucleus. Okay, because sometimes during the prophase also, the nucleus is still there. But supposingly, when once the nuclear membrane fragment, right, nucleus will be gone anyway. Okay, so that's why sometimes it's confusing. But normally I don't use nucleus as my point to actually identify. I use the chromosome. Okay. Then from metaphase, you can see it becomes thicker line. You can see compared to this one, it's thicker line. Okay, thicker line. So again, you see sometimes the nucleus is still there, but it's supposed not to be there. Okay, so that's why I don't use nucleus to identify. Okay, so this is I only look at chromosome, thicker line, and then you can see the shape start to spread. Okay, then compared to this one, metaphase and prometaphase. This one I think big difference. Okay, because this one, well, obviously you can see a line here that connecting. You can see a line that connecting like this. Okay, and there's space in between that. Right, then anaphase. Anaphase, sometimes this one looks like clumped together, but you can always compare with others. Because it totally depends on the staining of the cell also. Okay, so this one, it looks like telophase as well, right? Okay, but then this one see the line, that means that is still fluid. So this is anaphase. Okay? This one is totally come together. And this is the one that I said of the cell plate. You see that? The cell plate. Okay? And then this one is when certain part you can see it start to blur. Why blur? Blur because of what? Because of the chromosomes start to decondense to become the chromatin. Okay? So normally chromosome behavior, nucleus behavior is the 2 points that I use to identify it. Okay. Then next will be meiosis. Okay, for meiosis, right? Yeah, for meiosis, this one is the process where one diploid cell forms 4 haploid cells. So please remember, in meiosis 1 diploid to become 2 haploid. Then in meiosis 2 is 2 haploid become 4 haploid. Okay, that means that right after the meiosis 1 is already becoming the haploid cell already. Okay, and meiosis is important because it is actually producing the gamete, and meiosis is considered as sexual reproduction because it actually improves the genetic variation. Genetic variation is being done due to the crossing over that occurs and also independent assortment. Later I will talk about this. Okay, so that's why they can allow genetic variation due to crossing over and also independent assortment that occurs during the meiosis. Technically, the meiosis 1. Okay. So in meiosis, meiosis is important, right? Because first thing, they produce the gamete. They create the genetic variation. And one more very important thing is that they halve the chromosome number. Yeah, they halve the chromosome number. That is very important. For example, like human beings. Human beings, we have 46 chromosomes. So if let's say your mother don't do meiosis, they do mitosis to produce the gamete. Then what will happen is that the ovum from the mother will have 46 chromosomes. The sperm from the father will have 46 chromosomes. Then you will be having 92. Then you become a horse already. You won't be maybe a centaur, is it? Half horse, half man. Okay? So different chromosome number means that you will be different species already. You won't be human beings. Okay? Then later you and your wife will produce a son that is 184 chromosome. That one I don't know what, alien or something. It's impossible. Alright? So that's why you need meiosis. When you are doing meiosis, then you will halve the number of the chromosome. So that your mother will give you 23, your father will give you 23, then only then you will be just like human, 46. Then you will be having the same chromosome number like your parents. And only then, yes, you will remain as a human. Okay, so that's why it's important to halve the chromosome number. So that's why meiosis is important. Okay. So now meiosis, well, there are meiosis 1, meiosis 2, and the process, same. Prophase 1, then metaphase 1, then the anaphase 1, telophase 1, then prophase 2, then the metaphase 2, anaphase 2, telophase 2. That's the process. That's the step. Okay. So we will look at it one by one. Same thing in these 3 things: the chromosome behavior, the nucleus behavior, and the spindle fiber behavior. Okay. And this one we can go very fast. Why? Because only prophase 1 is different. The rest is all the same. The rest of it is all the same like the mitosis. So later you just can copy paste. Okay? Alright, only prophase 1 is different. And then there will be some slightly different in the terms that we use. A small difference in the terms that we use when we compare mitosis, meiosis 1, and meiosis 2. In other words, the terms that we use in the prophase 1, prophase 2, and the prophase 1 is slightly different. So that's why that one I will highlight that. Right? So you can highlight that in your table later. Okay? And that is the key point. That's the key point that you need to know. Okay, when it comes to comparison. Because some students they will get confused. Okay, why suddenly I use this chromatin? Suddenly I need to use the term chromosome. Suddenly I need to use the word homologous chromosome. Okay? Right, so let's look at it. I will highlight it later, so don't worry. Yeah? Okay, so the whole meiosis, it will be meiosis 1. So from 1 cell, it becomes 2 cells. Same thing, it has to start with the interphase first. Interphase, then meiosis 1. Then one cell becomes two cells. Then the two cells has, before they can undergo meiosis 2, the two cells has to undergo the so-called interkinesis, or another name called interphase 2. During this time, interkinesis or interphase 2, this is where the cell will grow bigger, increase in the protein and also the organelle in it, so that they can be further divided. Okay? So during this time, please remember there's no replication of the DNA. Interkinesis, when they are going for meiosis 2, there is no replication of the DNA. So after they grow bigger in size, and then only then they continue with the meiosis 2. Then they continue with cytokinesis. Then they continue. So that means that over here, there is also cytokinesis also. So that means that it will from one cell become two cells, two complete cells. And then only undergo interkinesis. Okay? Then only undergo meiosis 2. Okay? So it starts with interphase. So in interphase, everything is the same. During this time, nucleus is still intact. The DNA is still existing as the chromatin. They haven't condensed yet. It is still chromatin. Okay, now we start with the prophase 1. Prophase 1 in meiosis. In meiosis, you can see there are so many phases, right? Yeah, prophase 1 is even longer. Even longer than the prophase of the meiosis. You can imagine so long the meiosis, so many phases, it takes up to 80 to 90% of the time. So prophase 1 is where a lot of things is occurring. Most of the things all occurs in prophase 1. Okay? So same thing again, it is so long, the prophase 1 is so long. So that's why there will be early prophase 1, mid prophase 1, and late prophase 1. So in fact, mid to late prophase 1 is actually considered as prometaphase. It's just that most of the books, right, they don't want to further confuse you. So that's why they don't put prometaphase. They will just put early prophase 1, middle prophase 1, late prophase 1. They will just put like this. Okay. So next, chromosome behavior. Okay. So in early prophase 1, chromosome behavior, say from chromatin, it started to condense to become the chromosome. From the chromatin, it started to condense to become the chromosome. That's the one. Nucleus behavior, same thing. The nuclear membrane will start to fragment. Okay. Then spindle fiber behavior, they are just started to form. The centrosome are started to move to the different form. That is in early prophase 1. Then in mid prophase 1, in middle prophase 1, one special thing occurs. In mitosis, after the chromosome is being formed, they are just left. Okay? But in prophase 1, in the middle prophase 1, after the chromosome being formed, they will start to find each other. They will pair up. Okay? So the homologous chromosome will find each other and become a pair. So chromosome 1 and chromosome 1, they will find each other pair. Chromosome 5 and chromosome 5 will pair each other. Chromosome X, they will try to find chromosome X or chromosome Y to pair with each other. So all of these homologous chromosomes will pair up, and this process we name it as synapsis. Synapsis occurs. Okay. Then in the late prophase 1, because just now synapsis occurs, so the homologous chromosomes they will pair together already. So when the chromosomes they pair together, homologous chromosomes pair together, this pair of homologous chromosomes we will call it as tetrad or bivalent. Tetrad or bivalent. Okay, normally I use the term tetrad. Okay, so they pair. This whole thing is called tetrad. Okay, so in the late prophase 1, the tetrad, right, they will do the crossing over. Before this is like this, mid prophase 1 is like this. They just pair synapsis. Okay, so now during the late prophase 1, crossing over occurs. They will crossing over. So crossover occurs means that they will exchange the genetic material. They will exchange because this one comes from the father, this one comes from the mother. So they go pair, then they cross over. So when they cross over, this is where they exchange the genetic material. Okay. So this is what happened in the early prophase one, middle prophase one, and late prophase one. Okay. How about nucleus and spindle fiber? It's the same for the prophase. So nucleus during this time, middle to late prophase. Well, during this time, the nuclear membrane fully disintegrated and disappeared. During this time, spindle fiber has been formed, and spindle fiber has started to bind to the tetrads here. And during the crossing over, they have already attached to this chromosome. Yeah. Always happen, but it happens randomly. So we have for this 23 pairs of the chromosome. So sometimes it will be the upper arm will crossing over. Sometimes it will be this one and this one. Not necessarily this one and this one. Sometimes it will be this one and this one. Sometimes it's at the bottom one they will. Okay, so crossing over occurs totally randomly. So that's why you and your sibling, right? Even though it's from the same parents, but it still looks different. That's the reason. Okay, so I can show you here. You can see this, right? Okay. So this is synapsis. The process is called synapsis. Yeah, process is called synapsis. So they will form a tetrad. This is a tetrad. Okay. Synapsis is the process. Tetrad is the pair of the homologous chromosome. Okay. Then crossing over occurs. They exchange the genetic material. You can see here. Okay. So you can see here when one crossing over occurs, it will enable the genetic variation. How? For example, your father. If let's say your father is, let's say, black and straight hair. Black and straight hair, not black. Okay. So you see, black and straight hair. Black hair, straight hair, both of these are dominant. Okay. So let's say your father is like this. Black and straight. Okay. Then let's say your mother is having blonde and curly hair. The blonde hair is the natural one, not the artificial. You go and dye it. It's just different. Okay, so mother, let's say, is blonde hair, and then is having curly hair, blonde and curly. Okay, so if there's no crossing over, then that means that all the offspring will be either black and straight hair or blonde and curly hair. Okay, but because of crossing over, after they do the crossing over, right? So you see crossing over. So they exchange the genetic material. So it will become black and straight. This one will become the blonde and straight. This one will become black and curly. This one will become blonde and curly. So later on, each of these sister chromatid, it will be going into the daughter cell. Because you can see here, it will become 4. All right? So each of these will go, this one, black and straight hair, it will go into one gamete. This one, blonde and straight, it will go into another gamete. This one into another, this one into the 4th one. Okay? So it will produce 4 types of offspring. It can produce an offspring that is having black and straight hair. It can produce an offspring that is having blonde and straight hair. It can produce black and curly or blonde and curly. There are different chances. So that's why it increases the genetic variation. So you can see that just one crossing over, it can produce 4 types of it. So that's why it increases the genetic variation. And you know what? The crossing over don't just occur once. It don't just occur once. You can see there. This is under the electron microscope. Yeah. One tetrad can have 1, 2, 3, 4, 5 times of the crossing over at different parts of the chromosome. Everything is different. So that's why all the sperm, all the ovum, they are genetically different. Their pair are different, it's totally different. And somehow we have 33 pairs of chromosomes. One pair, let's say 5 different, 5 crossing over. It will create totally different. Okay, so that's why you and your siblings totally different, look different. Alright, that's the reason. Because there are so many things. Some of your siblings may be having curly hair, some of your siblings may be having straight hair. some of the sibling may be very tall, some of the sibling may be very short. It all depends on what is being crossed and what is being sent into the gamete. Okay, so that's how it creates the genetic variation. Okay. All right, then next, after that, it will be metaphase 1. Okay, so for metaphase 1, well, this is where the tetrad will be aligned at the metaphase plane. Okay, that's the difference. Because in the metaphase of the mitosis, it is the chromosome being aligned, right? So in the metaphase 1, meiosis 1, it is the tetrad, it's the homologous chromosome pair being aligned at the metaphase plane. That's the different thing. Okay, so yeah, I forgot to show you the paper. Okay, so for prophase 1 just now, you can see here, there will be early and late. Okay, so the terms that's being used here is different. Okay, so this one, first one, yes, chromosome is still the same. The early one is still the same, like the mitosis. Okay, so the difference is this one, after the synapsis. After synapsis occurs, they will produce a tetrad. Tetrad is actually a homologous chromosome pair. Okay, so during the late time, it is a crossing over that occurs between the homologous chromosomes. Okay, then the rest is the same. The behavior for the nucleus and behavior for the spindle fiber is the same for the mitosis. Totally the same. Only this part is the different. So you can highlight that. The terms that you use to describe is different. In prophase, you will state chromosome. In prophase 1, you will state tetrads or homologous chromosome pair. You use the wrong words in not that, so that's why you have to use the correct words. Okay. Then metaphase. During metaphase, what is being aligned? In metaphase mitosis, chromosome is being aligned. But in the metaphase 1, it's the tetrads or homologous chromosome pair is being aligned at the metaphase phase. Okay. Nucleus behavior, spindle fiber behavior is the same. So this one is just direct copy. Okay. Then in anaphase. In anaphase 1, what is being separated? In anaphase 1, it is the homologous chromosome pair being separated. So it's the homologous chromosome being pulled to the different pole. So in the picture, this is one of the key. This is one of the key. In the picture, you can see that it is what? It's a butterfly. Butterfly being pulled. Okay? Compared to the anaphase just now. The anaphase, what is being pulled? It's a fly. Okay? It's a fly being pulled. Compared to the anaphase 1 just now, anaphase 1 is a butterfly being pulled. So that's a difference. Sometimes they will give you this picture and then you have to know this one is it anaphase or anaphase 1. You need to be alert on that. Okay, that's how you differentiate it. One is a fly, one is a butterfly. Okay, so this one you can highlight also. Right, meiosis you can go very fast because this one is just repeating only. I just have to point out what's the different thing. Okay, so that's anaphase 1. So anaphase 1 is a butterfly. Okay, so this is the homologous chromosome being separated. And this is anaphase 1. So you see here. You can say tetrad being separated, or you can say homologous chromosome is being pulled to the opposite pole. The rest is the same. Okay. Then next, telophase. Telophase, the homologous chromosome will reach the opposite pole, and then same thing. They will start to decondense. They will start to uncoil back to become chromatin. Nucleus will be reformed. So it will reform the nucleus. It will still undergo cytokinesis so that they can fully form two cells, two different daughter cells. Okay? Then these two daughter cells that's newly produced, right, they will be very small. So that's why they have to undergo interkinesis or the interphase 2. So during this interphase 2, they will only grow bigger in size only. Okay, they won't do the DNA replication. Just remember that. Okay, so this is the process. So in the end, it will still form two cells. They will still form the nucleus. Those chromosomes will still decondense to become the chromatid. This is all the thing that you need to know. Right? Because some students, they were confused. They were thought that, oh, no need already. It will still, right, form a normal cell first. Then the cell will undergo interphase 2 or the so-called interkinesis. So please remember, during this time, they only increase in the size of the cell. They won't do DNA replication. They won't do DNA replication. Okay, then next, we continue. Only then the two daughter cells, they grow bigger size already. Then only then, they continue with the prophase 2, metaphase 2, anaphase 2, and telophase 2. All of these two, meiosis 2, is exactly same like mitosis. The terms that's being used is exactly same like mitosis. Okay, so that's why you can see here, in prophase 2, in prophase 2, same thing. Chromatid start to condense to become a chromosome. Okay, so during this time, there's no synapses, no crossing over area. It's totally same like the prophase in the mitosis. Okay, so that's why in prophase 2, here is the same. Everything is the same. So you copy from the mitosis. You copy from the mitosis. Okay. Then metaphase 2, same thing. Metaphase 2, in the metaphase of mitosis, it is the chromosome being aligned at the metaphase plate. Right? So over here is also the chromosome. What is the difference? Because when they want you to compare meiosis 2 and mitosis, what is the difference? The whole thing is the same. You can see that the term that is being used is the same. Everything is the same. But what is the difference? The difference here is that the chromosome number, half of it. Okay. So for example, if let's say they draw not 6 chromosomes being aligned. Then another picture they draw only 3 chromosomes being aligned. Okay. So in this case, which one is the metaphase? Which one is the metaphase one? Or metaphase 2? You need to be very clear. Okay. So based on picture, sometimes they will use picture to ask. Right? So that's the thing. The major thing. Chromosome number will be just half of it. If you will ask you to choose which picture is the metaphase, which picture is the metaphase 2, which picture is the metaphase 1, they will ask you to choose something. Right? Yeah, this kind of question. So that's why you need to be clear on that. But the terms to describe it is the same. It's the chromosome. Okay. Then for anaphase 2, same thing. It's the fry that's being separated, right? It's a fry that's being separated. So that means that it's what? It's a sister chromatid being separated. Same like mitosis. Right? In mitosis, anaphase is also the sister chromatid being separated. So it's the same. So how to differentiate between the anaphase and anaphase 2? How to differentiate it? Well, the sister chromatid is both of them are separating the sister chromatid, but the sister chromatid, their genetic content is different. In the mitosis anaphase, the sister chromatid is being separated. All of these sister chromatids are genetically identical because no crossing over occurs, so they are genetically identical. But for the anaphase 2, the sister chromatid is being separated, right? But you see the sister chromatid different color. Means what? Means that the sister chromatid that's being separated here are genetically not identical. They are not the same. Why? Because before this crossing over occurs, right? So that's why their genetic content is different. Okay. So that's a major difference that you need to highlight. If, let's say they ask you to compare meiosis 2 and also the mitosis. Okay. So that is for the anaphase 2. Then lastly, telophase 2, everything is still the same. Okay, so it is basically the sister chromatid reach the opposite pole, so they will start to decondense, uncoil to become the chromatid again. Then the nuclear membrane will form and blah blah blah. All this same, totally same like mitosis. So that's why then after that cytokinesis occurs again, then the cell will become another 2. This one also become another 2. So total 4 cells being produced. Okay. So in the table, you can see that everything is the same. Everything is the same like mitosis. But what I highlight just now is in the prophase 1, in the prophase 1, the number, right, or the metaphase 2, prophase 2, the number of the chromosome, that's the first thing that you differentiate from the mitosis. Second thing is anaphase 2, the sister chromatin. Mitosis 1 is totally identical, it's the same one. This one will be different. This is a 2 point that you need to know. Even though you look at this, so the whole table here, prophase 2, so all the meiosis 2 one is totally same like mitosis 1. But then, that is a different that you need to understand. Okay? So that would be the telophase. Yeah, so with that, that is basically all about the meiosis process. Okay? So, done, then one last thing is about the comparison. Yeah, comparing the mitosis and meiosis. Comparing the mitosis and meiosis, there's 3 major points to compare. Okay, 3 major points. Yeah? So the first major point is that we will talk about the number of the cell that's being produced. Number of the cell that's being produced. So mitosis is 1 cell produce 2 daughter cells. Meiosis is 1 parent cell produce 4 daughter cells. Okay, that's the first one. Second point is to look at the genetic content. Genetic content for mitosis, it is going to produce diploid, Okay? Meiosis, 1 diploid cell. After meiosis 1, it becomes the 2 haploid cell. Then meiosis 2, 2 haploid cell become 4 haploid cell. So it's diploid become haploid, haploid become haploid. Okay? So that is the second point. Then the third point is, of course, the whether it's genetically identical or not. So the offspring, the daughter cell, will be exactly genetically identical to their parent in the mitosis. But in meiosis, their genetic content will be different from their parent. Okay, so with that, yes, that is basically the comparison. And that's all for this part of lecture. Easy and simple. Yeah, easy, right? It's something that you have learned before, and then everything can be summarized. So that's very easy. Okay. So on this Friday, we will continue with the lecture part 2. Yeah, the transcription, replication, transcription. I will continue with the lecture part 2. Okay. So with that, that's basically all. Remember to do the extra question and do the revision. And the test is just next week. Yeah. Okay, so basically that's it. Anyone haven't signed in your attendance, please remember to sign in.